The first speaker today is Professor Margaret Gardell of the University of Chicago. At the ripe old age of 32, she has numerous awards, one of course being a Sloan, a Packard. She was a Pappalardo Fellow. She was also a Jane Coffin Child Fellow at Scripps Institute. She has a Burroughs Wellcome Center Career Award. And I could keep going on. The most, at least the most amount of money award is a recent NIH Director's Pioneer Award, which is essentially $2.5 million over the next 5 years. So Margaret, I guess in terms of all the awards, I should also say that she had a PhD at Harvard, and then went on to do postdoc at MIT, and then, of course, Scripps, as I mentioned before. So without further ado, we're very happy to have Margaret talk to us about building contractile bundles for cell adhesion and migration.
Well, thank you very much, Jen. I myself am very excited to be going early in the day, so I don't have to, go after Jean-Francois. The field I work in was nicely introduced by Jean-Francois and Jacques last night. It's a field of understanding how protein dynamics in cells give rise to their mechanical behaviors and migratory response. And today I'm going to speak of a problem we've been starting to work on. It's all unpublished data of understanding how cells build contractile networks in bundles that mediate forces that cells generate on their extracellular matrix. So this is a movie of an endothelial cell migrating through a three-dimensional collagen matrix. You can see, and it's a phase contrast microscope image. And the collagen fibers you can see nicely outside the cell, these phase-dense regions. And you can see as the cell sends out a protrusion, it contracts that matrix, which requires a force. And at the rear of the cell, it lets go of these collagen fibers and detaching, thus releasing the tension that was imparted on this extracellular matrix. Cell migration, this process of cell migration is important for many different aspects of development and sort of maintenance of our physiology, including wound healing and extracellular matrix remodeling during development. And of course, when it goes awry, it can result in tumor metastasis. So we can think about the mechanical aspects of cell migration as a cyclic one that involves the mechanics of cell protrusion, the mechanics of adhesion and force generation, and then detachment at the cell rear and changes in cell shape that allow the cell rear to retract and for processive movement. If we look inside the leading edge of the cell, we can use green fluorescent protein tagged to actin, the filament structure that I'll be speaking of today. And so this is a GFP actin image taken with confocal microscopy. And you can start to see some of these bundles and networks that are formed inside the cell. And much has been learned about these networks through numerous molecular biology and biochemistry studies. And our picture of this leading edge of the cell is that there's this lamellipodia network at the leading edge, which is the protrusion is mediated by polymerization of actin filaments that are cross-linked into a dense dendritic array. And this is what allows cell shape change at the front of the cell. Immediately proximal to the lamellipodia are these contractile bundles. These contractile bundles are formed from a motor protein, which I'll speak of more in detail. And so the formation of these contractile bundles are myosin 2 dependent, and there's a specific isoform of myosin that's important for this. It's myosin 2A. And then a network that you can't see very well by light microscopy, which is what is essential for maintaining the cell shape, for making it form these convex or concave shapes. Is the cellular cortex, which is you can think of as a two-dimensional network, isotropic network of actin filaments cross-linked with cross-linking proteins and other motor proteins. And the actin cytoskeleton is linked to the extracellular matrix via these adhesion sites called focal adhesions. And as a physicist, I like to simplify these to one protein. I sort of compromise with a cell biologist, so I think of about 4 different proteins. But in reality, there's about 100 different structural and signaling proteins that mediate the interaction between the actin cytoskeleton and the extracellular matrix. Quite generally, you can sort of classify these proteins as either actin-binding proteins, these core proteins that usually bind to proteins that bind to actin, and then to plasma membrane inserted proteins, and proteins, these ECM-binding proteins that bind directly to the extracellular matrix. So we can think of the focal adhesion as existing of layers of proteins that mediate the transmission of forces between the actin cytoskeleton and the extracellular matrix. So the mechanics of how forces are transmitted through this dynamic assembly of many proteins is one that in recent years has really been advanced by advances in light microscope techniques such as, which you may have heard of, such as PALM or STED microscopy. So the motor protein that we're thinking of is myosin 2. And myosin 2 in vitro is a dimer, and each of these are its motor domain heads, which are enzymatically active. In the cell, you don't find individual dimers of myosin very much. Instead, these motors self-assemble into these bipolar mini-filaments. So you have orientations of heads pointing on either side of you, pulling in actin filaments or translocating actin filaments towards the center of the myosin 2 mini-filament. And many of you may know this already, but myosin motor proteins have a force-dependent velocity. So if there's no load imparted on its substrate, and here, actin filaments for the case of myosin 2, if this were a processive motor, it could move the actin filament at a speed of about 100 nanometers per second. In the cell, there are loads imparted on the actin filaments, so even a force as low as 2 to 5 piconewtons can install this motor, and so then you can transmit tension through this actin-myosin interaction. And in fact, the binding affinity of myosin to the actin filament is load dependent, so it actually stays bound longer to the actin filament the more load that is imparted on that bond. So in the cells that we're thinking of, we want to know the role of myosin 2 in building these contractile bundles that we see here by immunofluorescence. This is an immunofluorescence image of the actin cytoskeleton in green, the focal adhesions in red, and you might be able to see them, but it's hard to see in the color combined, the myosin 2 mini-filaments which decorate these actin bundles in blue. And so the way we study this assembly process is by using a myosin 2 ATPase inhibitor called blebistatin. When you treat cells with blebistatin, it binds tightly to the ATP pocket of myosin 2 and prevents its tight binding affinity to actin. So when you treat the cells for 30 minutes with blebistatin, you can see that all these bundles are dissipated. You just see a nice fine actin meshwork left behind. And you'll also notice at this point that the actin focal adhesion puncta are limited to the leading edge of the cell and right at the base of this lamellipodia network. For those of you that are interested, the transition, this is still the lamellipodia network that's completely polymerization dependent. If you treat the cell with blebistatin, that lamellipodial network is unaffected, and you still see the formation of small focal adhesions at the base of that lamellipodia. Now this process is reversible. So now if under the microscope we wash out this drug, we can see within 15 minutes we can reassemble these contractile bundles. So the fact that we can, under the same experiment, inhibit this bundle formation and then reassemble it within reasonable timescales accessible to us by the light microscope allows us to watch the self-assembly of these bundles in real time. So this is a question that Yvonne Aretin, a cell biologist in my lab, has been studying for the past year and a half, and it's her results that I'll be presenting. So what Yvonne does is express green fluorescent protein conjugated to actin and simultaneously with a red fluorescent protein, MAPL, conjugated to paxillin. So this is a UTOS osteosarcoma cell where she's expressed both these constructs as imaged with spinning disc confocal. If she treats the cells with blebistatin for 30 minutes, you can see those bundles have now all dissipated. You see some sort of fat, wavy bundles in the center of the cell, and you can see nicely these small punctate near the cell periphery. And now at time 0 is when she's washing out the blebistatin. So we're going to watch the reassembly of these bundles, and this movie is sped up quite significantly. The timestamp here is in minutes and seconds. So as she washes it out, you can see within the course of 30 minutes, you see the reassembly of these bundles, the elongation of these focal adhesion plaques. So she can actually visualize the coarsening of these actin-myosin bundles. And we're interested in the forces that are generated by these bundles. So she doesn't plate these cells on a rigid cover slip. Instead, she plates the cells on a fibronectin-coated hydrogel, polyacrylamide, that's embedded with a fiducial markers that allow her to visualize the strain field inside that gel. So this is a technique called traction force microscopy that was developed in the mid-90s by Yuli Wang and collaborators. And so now, by putting beads in the third color, she can visualize actin and focal adhesions and also fiducial markers. These are actual images that actually I took in a different cell type. This is actin. These are the beads. If you, at the end of our experiment, we have to see the relaxed state of the gel, so we trypsinize the cell to detach it, and you can see, after detachment, before detachment, you can see the deformation of those beads. At the end of the experiment, the cell is completely gone. So now we use particle imaging velocimetry data to track the deformations of the beads and get a strain field. So on the left here is the strain field of the beads in the matrix that we calculate. We usually measure about 5,000 strain vectors. And in collaboration with Ulrich Schwartz's group, who's now at Karlsruhe in Germany, a master student in his group, sort of modified some traction force reconstruction algorithms to calculate the traction stresses. Basically, we know how stresses propagate in our polyacrylamide gel. It's a semi-infinite elastic half space, and then we can use the strain fields to then make an estimate of the traction stresses that the cell is imposing on it. So this is our final our stress vector field. With the density of strain vectors that we use, we can obtain a stress vector field of approximately 1 to 2 micron spatial resolution and a force or stress resolution be from 10 pascals, which is the forces generated on the substrate by the lamellipodia network, all the way up to several hundred pascals, which is that generated by large contractile structures.
This is a 3D image or 2D?
This is a 2D image, so we're imaging the top surface of the gel.
So you're only learning then about strains, not no normal?
No, we don't know any normal strains. I mean, we could in practice do that. Except, as I was speaking with Paul about, on soft matrices we see a lot of normal strains, but on these rigid surfaces, I mean, the strains are on the order of a micron. So it's starting to be within the resolution of our ability to measure Z and with confocal. But that would be also interesting.
There are shear stress also, right? Do you measure shear stress?
Shear stress. So we're measuring the surface stress that's applied to the polyacrylamide gel, but tangential to the, we're only measuring the shear direction.
What is this stress and also normal stress, right?
So we're only measuring stress and shear that's parallel to the surface of the gel.
You do measure.
Yeah. Okay, so these are an example of our traction vectors that we obtain, and it's a little bit easier to see it so all the traction vectors always occur towards the center of the cell. You never see traction stress vectors going in any other direction. So this is the magnitude of the traction stress vectors, and they're all pointing along the length of these bundles. And the magnitudes of the stresses exerted at these bundles can range from around 40 pascals all the way up to 140 pascals. Okay, and if you don't like thinking about pascals, you can think about it in terms of piconewtons per micron squared, which is also a useful unit to think about when you think about how much molecular forces are required to generate these stresses. So now with this analysis of the traction stresses, we can look at the traction stresses simultaneously with the actin and paxillin dynamics. You can see early on in this movie, we see stresses that are very close to our resolution limit using this stiffness of polyacrylamide gel, but you can, at this point, you can just make out a faint boundary of stresses, but we'd have to go down to a lower elasticity of our matrix to be able to resolve these myosin-independent stresses but then we can watch the stresses develop as the focal adhesions enlarge and the actin bundles grow. So first of all, this is besides the analysis of the traction stresses, we can also quantitatively analyze these actin movies. And as you look at these movies, I would love input as to what kind of parameters you think are important for for predicting what kind of traction stresses we would measure. Because that has been actually the struggle we've had so far is figuring out what parameters actually are predictors of the magnitude of the traction stress. So the easiest correlation we've done so far is just take a point of one of these focal adhesions that elongates and results in a high traction stress and follow the length of the focal adhesion as a function of time and correlate that to the traction stresses. So here's a characteristic curve that we see over one of those focal adhesion spots. And again, time zero is always time zero when we wash out the drug. And so immediately you see this rapid increase of traction stresses, and then a slightly slower increase that then plateaus for a small amount of time, but then there's a slow increase after that as well. And this kind of trend is seen for focal adhesions on stiff matrices. There's been a lot of work suggesting that the focal adhesion length could be correlated to how much traction stress is being applied. However, we only see this is a measurement of focal adhesion length. We see that the focal adhesion tracks very nicely with increases in traction stress at early times, but that at later times the adhesion length saturates and does not change, where the traction stress can still increase slowly over time.
Margaret?
Yeah.
What is adhesion length?
Sorry, so we're measuring, so the width of the focal adhesions.
The size of those little dots?
Yeah, it's the size of it's actually the length measured perpendicular to the cell perimeter. And if you look over time, that length increases over time.
Thanks.
And so there have been models out there in the literature that have correlated large focal adhesions with enhanced forces. So actually different models predict whether, actually some models predict that the stress at the focal adhesion sites remain constant. I think our data never shows that the stress at the focal adhesions remain constant. We always see an increase in force per unit area. And also we don't always see a nice correlation between enhanced stress and increased focal adhesion size. That there is there's a time scale when you see a high correlation of this is just a correlation between traction stress and adhesion length. And in this fast phase, we see a very high correlation between these two parameters, but at longer times we see very little correlation. So the traction stress is not always correlated to the size of the focal adhesion sites.
This is for average data over all the focal?
That data, this correlation coefficient is averaged over like 40 different focal adhesions from 8 different cells. So so I show you characteristic data sets. Actually, later on I'll show you data sets that amazingly were able to be averaged from different data points. Yeah. So the width never increases. Yeah, so the lateral dimension doesn't change as it's growing longer in these cells at least. So the focal adhesion is like an oblong oval shape. The width of it does not change. It always stays constant from the beginning to the end of the movie, and it's this length that always increases and changes. Oops. Okay, so since we saw these different phases of traction stress recovery after washing out blebistatin, we broke this data set into two different time scales. There's a very rapid time scale where we actually are watching the initiation of traction stresses, the rapid buildup of traction stresses, and then we see a longer time scale, which we think is due to actin-myosin remodeling. So immediately after washing out the drug, and immediately I mean this is the velocity of the actin vectors between 0 and 7 seconds washing out the drug. And so here you see that the actin filaments undergo a very rapid retrograde flow towards the center of the cell. So in this region, you see all the vectors are pointing towards sort of this mid zone of the cell. And between at 20 seconds, this is on the same scale, these velocity vectors have decreased or diminished significantly. So if we look at the average retrograde velocity of these of the actin cytoskeleton in time after blebistatin, we see that immediately after washout, we see this very rapid retrograde flow that's diminished within 10 to 15 seconds after 10 to 15 seconds. And then this slower flow. It's above our resolution limit, so it's a stationary retrograde flow that's observed in these cells at around 0.2 microns a minute. But there's a very rapid flow and then stabilization of that flow that occurs very early on after washing out the drug. And what we were really surprised to see is that during this time, if we look at these focal adhesions underneath the cell, that at these time steps, these Paxillin spots, so this I'm looking at 0 seconds and 7 seconds, are actually translocating. So these adhesions that are mediating interactions of the extracellular matrix are actually moving and slipping during these times. So I'm showing you a color combined images between 0 and 7 seconds here, so you can see nice red and green that don't overlap. And then 7 and 13 seconds, you see that there's nice yellow indicating the focal adhesion spot has not moved during this time, and that it's simply elongating off the back of the this punctate structure. So we can know it's slipping because we can measure the velocity of the actin, and we can also measure displacement over those same timescales of the substrate because we have the beads in the substrate. And we can measure, we can show how this displacement rate, I hesitate to call the substrate velocity, but so I just think displacement rate for some reason sounds better to me, but it's the same thing. That this displacement over those timescales of the substrate is quite low, below a micron per minute, and the Paxillin slips very rapidly early on, but then it gets indistinguishable from the substrate. And that actin flow also then starts, and then immediately after this engagement of that Paxillin spot, the actin flow then becomes abrogated. So it's almost like this actin flow, the myosin motors start acting on the actin, pulling back this meshwork, and then as the focal adhesion sites become engaged, that actin network becomes stabilized. So you can just think about this, there's this shear happening between the actin network, these Paxillin, the adhesion spots, and the substrate. And then the Paxillin then these adhesions become engaged, but the actin can still shear on top of those adhesion sites. And all the while, we can observe that the traction stresses that are exerted onto the substrate are increasing during this slip time. So from before and after the adhesions stop slipping, the traction stresses exerted increase from 20 pascals up to about 40 pascals. So this is suggesting that those adhesion sites at the front of the cell are very labile, but they can still exert a frictional drag on the substrate, and that once they become engaged, then higher stresses can be exerted on the external matrix. So as I mentioned in the first slide, I'm not going to go into all the details, but there's several different layers of many different proteins within the focal adhesion complexes. For us, the most important was the interaction between the transmembrane protein integrin and the extracellular matrix. And it was quite surprising for us to find that this integrin, which is that last link between the inside the cell and the external matrix, also slips, and it slips at the same speed and concomitantly with the Paxillin. So now in these nascent focal adhesions, you have these transmembrane proteins actually slipping right with the whole focal adhesion complex and then becoming stabilized. So in these nascent focal adhesions, immediately as you're building up tension from myosin 2, you can think about this slipping clutch, which contains, vinculin, for those of you who care, vinculin, talin, Paxillin, and integrin all together, and then and it suggests that there's this critical amount of tension that's required to then stabilize these bonds and stabilize these bonds to the extracellular matrix to be able to transmit forces from the actin cytoskeleton. So, and I say that this suggests that there's a critical stress, but we can actually test to see if this really is a critical stress by modulating the spring constant of the extracellular matrix. So if we make the extracellular matrix softer, the cell will have to exert larger amounts of strain to exert the same amount of stress on those focal adhesion complexes. And so we can then test to see whether this process is strain controlled or stress controlled. Okay, so these are our results. We plated the cells on different matrices of different stiffness. This is this gel stiffness that we plated the cells on in kilopascals. So we've changed the gel stiffness by about a factor of 4. And this is the strain that's exerted in the substrate during this slipping time. And then if we look at the stress that's exerted, the stress remains relatively constant. So when we plate the cells on softer matrices, they deform that matrix more to be able to obtain a critical stress within that focal adhesion complex to be able to engage the adhesion site. And what that means is if it's deforming the substrate more, that also means the internal actin cytoskeleton also has to be deformed more because it has to actually move that substrate a larger displacement. So we can actually look at the amount that the actin cytoskeleton has contracted during that slip time. And this is on a rigid substrate, so the actin cytoskeleton is contracted by about 0.7 microns during that time. But then if we look on the soft matrix on these, the actin cytoskeleton is contracted almost already 2 microns. So there's more contraction happening in the actin cytoskeleton, and this is required to be able to get that critical tension within that focal adhesion complex to be able to stabilize those adhesions. And since we're looking at the velocities between these different layers, we can actually look at the sort of get an estimate of what the frictional drag is across these layers. Because we see a shear, we can measure the velocity. We can, by a simple force balance, we can say that the forces across these interfaces are equivalent to each other. And so we can get just a rough estimate of the compliance. I mean, obviously, this is measured at a certain frequency, so we're measuring the compliance at this frequency. But we can just get a rough estimate of the relative velocities divided by that force between the actin and the focal adhesion interface and the focal adhesion and the extracellular matrix. And so if we look at this effective viscosity, this is our resolution limit, our ability to resolve differences in displacement. And on the soft matrices, you see that this is time again after blebbistatin treatment. Initially, this viscosity is very low between the actin and the focal adhesion, and then increases by about an order of magnitude within 25 seconds. That means the frictional drag between the actin and the focal adhesion complex is changing during this time. But remarkably, when you look at the drag between the focal adhesion and the extracellular matrix, this actually increases by roughly 2 orders of magnitude in that same time scale. Right, so that drag between that integrin and that extracellular matrix is increasing quite rapidly, which is consistent with what we see by eye of this, engagement of these focal adhesion complexes. And if we then compare these changes in effective viscosity between the soft and the stiff matrix, on the stiff matrix we see very similar increase in rise between the actin and the focal adhesion complex. But we actually see a more rapid rise between the integrin and the ECM. So that suggests that there's some, stiffness-dependent strengthening of that integrin extracellular matrix bond that happens a bit more rapidly on the stiff matrix than the soft matrix. Okay, so what we've identified here is that these initial adhesions that when in the absence of blebbistatin, you form adhesions that are very weakly coupled between the actin cytoskeleton and the ECM, and this sort of makes sense because if you're a cell exploring your new space, you don't want to get permanently stuck to all these points that you're exploring with your lamellipodia network. So these weak adhesions are very labile. Other groups have found that they turn over very rapidly, so they assemble and disassemble rapidly. And then what we find is that there's this critical amount of tension that needs to be applied to these weak adhesions that will stabilize the bond between the integrin and the extracellular matrix. So we see that the myosin 2 forces drive this rapid increase in strength between the integrin and the ECM, and that really does seem to, this part of the traction force buildup, it seems to be stress controlled. Okay, so next we're gonna go everything after 40 seconds, when now we've stabilized these adhesion sites, and so here we're going to look at how the myosin is remodeling the actin cytoskeleton to build these bundles. So here Yvonne has labeled myosin light chain conjugated to green fluorescent protein, and you can see this is a control situation. So you can see these nice myosin, actually you can visualize the myosin mini filaments by light microscopy. These are those punctate structures that you see along the bundle. And if she treats the cells with blebbistatin, blebbistatin does not affect the assembly or disassembly of these myosin mini filaments, so you can now just see that these myosin mini filaments are disorganized throughout the cell. Okay? And as she washes out the blebbistatin, now you can see as the myosin motors are reactivated, the compaction of these bundles and coarsening into these contractile structures. And so if we look at this at different time points and correlate sort of what the structure looks like at different time points, initially you see a quite disorganized network, and at 2 minutes, your stress is already increased to about 2/3 of its maximum value, but you still don't see organization of these very compact bundles. But by 24 minutes, at the end of our experiment, we see a nice striated localization of these myosin minifilaments along actin bundles. And I should say that because these myosin minifilaments are always pulling actin filaments contractile, we know that it's been shown that the actin filaments are not spanning the full length of these bundles. They're actually, still 2 micron. They're about the length of the spacing between myosin minifilaments in this picture. So this bundle is consisting of these short actin filaments and myosin mini-2 cross-linking them, minifilaments cross-linking them, and generating force on them. So if we, again, look at the retrograde flow of the myosin, we see the same exact thing that we saw with the actin, and Yvonne's actually shown that the rate of retrograde flow of the myosin is equivalent to that of the actin. So immediately you see this very rapid flow, and now I started plotting things in terms of nanometers per second just to be consistent with for some reason I did it. I don't know why. And so immediately after watching it, you see this very rapid flow. And what I like to look at in this movie is that you actually see, places where there's a sink. So you see flow going to a certain point. You don't really see divergences so much. You mostly see sinks and places where flows are coming into a central point. This flow quickly stops. Within a minute after the washout, even before that. And this is the steady state flow. And what happens at the steady state, you actually see correlated motions of the myosin minifilaments along a bundle. And other people have observed that myosin minifilaments can actually expand and contract along that bundle as well. So there's some correlated motion of entire bundles, but there's also expansion and contraction. So the fact that we see this change in speed at very early times led us to wonder if we could just look at this as a simple force-velocity relationship that is known for myosin 2 in vitro. And so if we plot now the actin or the myosin speed as a function of traction stress, we see a nice, with the amount of data we actually have, an inverse correlation between these two parameters. And so if you can explain the load-dependent velocity of either the actin or the myosin as the unloaded velocity times 1 minus the ratio of the stress that's being pulled on it divided by the stall stress. And so if we then calculate the slope and y-intercept, which gives us these parameters of the unloaded velocity and the stall stress, you can see that on our different matrices, we actually get unloaded velocities that are the same order as those measured for in vitro experiments. And even stall stresses, depending on what area we use for converting stress to force, that are also similar to single myosin 2 motor proteins. So this suggests to us that initially what's happening is you're sort of pulling, you're rapidly contracting this network, and this changes in velocity that we can see is just because these myosin 2 motors are getting more loaded by stresses that are being exerted to it by the substrate. However, so this is the data set that we see at very early times, and so we have about 5 data points there. When we look at later times, there's a constant retrograde flow, and so we see a very nonlinear force-velocity relationship at any times or any forces above 40 Pascal. And so at between 40 Pascal and 120 Pascal, that's when you see this coarsening from this sort of disorganized meshwork to more organized bundles. And we don't have any way of explaining the nonlinearity in this force velocity or why these networks generate different amounts of tension on the extracellular matrix. But that's what we see. So then the question is how does this organization of these myosin minifilaments? We saw a big dependence on external matrix parameters when we plated the cells on stiff versus soft matrices. We saw a very big difference in how their adhesions engage themselves to the extracellular matrix. So it's been well studied by Paul Jamny and other groups that when you plate adherent cells on matrices with different compliance, you actually see a very different morphology of their adhesions. So on soft matrices, you see very small adhesions that are dispersed throughout the cell. And on stiff ones, you see very numerous adhesions. And you also see fewer actin bundles. It's not necessarily that evident from this image, but you don't see, and as you'll see, you don't see as organized actin bundles. However, we're really surprised to see, so previous measurements have looked at the integrated traction stress across the whole cell. So if you integrate the traction stress and compare the soft versus the stiff, you'll see that the amount of traction stress the cell exerts scales as the stiffness of the substrate that is plated on. So the traction stresses exerted by this cell integrated are roughly about 4 times less than the traction stresses on the stiffer matrix. But you still see points where the traction at these engaged focal adhesions, the stresses can still be quite high, much higher than we were expecting. So local stresses can be high, but the overall stresses are lower. So on the soft matrix, we see, this is our lovastatin washout movie. We see that at the beginning of the movie, you see this massive compaction of the cell. And you do see a reorganization into bundles. You see the lamellipodia network, but most importantly, you see this massive deformation. And if we correlate the similar to what we did on the stiff, you can see that over time, the soft one doesn't really organize these compact bundles as well at longer times. But there is very rapid retrograde flow. And what was really surprising to us is this is our traction stress as a function of time on the stiff matrix. And what we actually observed at our single focal adhesions is that the traction stress actually increases much more rapidly on these soft matrices, but then plateaus. So you don't see that long time scale coarsening of these networks, but you see a very rapid increase in traction stress. And what we found recently was that if we instead of plotting it as a function of time, but plot the traction stress exerted as an amount of total network contraction, so the amount that the actin or the myosin has moved from the beginning to the end of the image, so that the total contraction, we actually see that on these 2 different matrices, you can predict the traction stress by the amount of, not necessarily deformation, but contraction of these 2 networks. So it's like you're building up some strain inside the network, and that is telling you how much traction you're getting, at least until this 80 Pascal limit. And then we still don't understand that long time scale coarsening. Since I'm being yelled at, I think I'll just go away. So we also did a little bit of looking at how alpha-actinin, a cross-linker, plays a role in these bundles. And I showed you that movie, so you don't need to see that movie again. But what was really interesting to us is that the alpha-actinin also shows this nice striated localization along these actin bundles. But in contrast to the myosin 2, its localization completely dissipates as you inhibit with levastatin, and its reorganization, its relocalization of these bundles is dependent on the reorganization of the myosin 2. So the localization of the alpha-actinins requires myosin 2 dependent remodeling. And we're trying to figure out ways of characterizing that. And it looks like you can think about this connectivity of these contractile bundles spanning the cell and sort of measure these connected networks of alpha-actin and myosin bundles that are spanning the cell over long time scales. So these are the clusters of close alpha-actin and myosin that are in close proximity to each other. And you can see that the number of large clusters increases after this washout. So we're just trying to look at the reorganization of these contractile structures. So this is nothing that I haven't said before. So we're trying to look at do live cell imaging and measurements of traction stress to understand how cytoskeletal proteins self-organize into structures that generate stress on the extracellular matrix. And I would love help from you to figure out what other parameters we can make lots of measurements of flow and deformation and stress, and we need to know what parameters are important to make predictions about these material properties of these networks. And all this work was done by Yvonne, Ayrton Schaus, and I think very much our collaborators who've provided fluorescent protein constructs and have worked with us on these traction stress algorithms. So thank you very much.
I'm just curious about so I like the idea of the whole focal adhesion assembly, like shearing and each component slipping or whatever, but the integrin, how do you envision that slipping? Is it that then the fibronectin's stretching or something, or how is that slipping relative to the substrate?
Yeah, I think that so there recently, people know that integrins can go in a weak conformation binding and a tight conformation binding, and that is a tension-dependent process.
Yeah.
So, I mean, right now we're doing the control experiments to check to see whether this can be done with simple RGD peptides. So if you need stretching of fibronectin, which I don't think is happening because we don't see much fibronectin remodeling on the matrices with these cells. But if we can see it with a peptide, then we know it's not stretching in the peptide. And we also will know with that whether we need the synergy site in fibronectin to be able to get this slip stick motion.
So when you say slipping, then do you mean you're including stretching and slipping?
No, I see it as this transient bond between the integrant and the fibronectin. That you have a certain percentage of them that are bound at any time, and that percentage of molecules that are bound increases as you go from the slipping phase, the frictional slip phase, to the stick phase. So it's not necessarily like a stretching.
Is it known what is the correlation between the amount of stresses and cell motility? So you show that on different substrates the total stress is different, probably for different cell lines is different. So what is the effect?
Yeah, so let's see. Usually cells that generate low stresses are more highly motile. So traction stress is it's biphasic. So if you don't have enough adhesion and can't exert any stress, then you can't move. But if you exert too much,
it has some
yeah. And so people have done that by looking at migration rates on substrates of different stiffness and been able to see that.
What did the cells do at boundary between a stiff and a soft?
So Yuling Wang found that they durotax. So if confronted with a boundary, they'll always move towards a stiffer matrix. Yeah.
When you do the trypsinization of the cells that have been plated for a long time to see which way the beads relax,
yeah.
Is there a correlation in the displacement vectors with myosin mini filament directions that you see? Because those also often seem sort of radial, and yet the relaxation stresses are all in.
Yeah.
Right?
So
are those correlated or anti-correlated?
let's see. So there's definitely a correlation with the direction of retrograde flow.
Yeah.
Okay. So but you're asking any
those dramatic active
bundles, the transverse
myosin
bundles.
things.
Yeah. There's not. I mean, those transverse bundles aren't linked to focal adhesions as much, so they don't actually exert that much stress on the substrate. But if you do see a correlation in strains, it's with the direction of the bundle.
But when you let a statin treat the cell, those things go away.
Yeah, but
those things are not responsible for the traction stresses on the
they're part of that connected network, but like usually the traction stresses are linked to bundles that are perpendicular to the leading cell edge, and those bundles hook up farther away from the substrate. So they contract, so they move this way, but they're also moving retrograde at the same time.
Which is not so evident in those fluorescence pictures.
The transverse arcs?
Yeah.
Oh no, yeah, you can see that. Let's see, different depending on your cell geometry. You can see different. So here, there's so these are transverse arcs, and then these are the bundles that are actually linked up to focal adhesions. You can sort of see if they pin these transverse arcs.
But it's the radial ones that are responsible for the relaxation you see after you detach the cell.
It's these.
Yeah, those.
Yeah, those. And these are not linked up to adhesion sites, but they're part of the network that is contracting the other bundles.
Okay, with that, we're going to have to move on to the next speaker. We're going to try to shave a few minutes off since we started at late time. So.